Assalamualaikum and very good afternoon to everyone. Today I will present about the research on the effect of its sample type and location on industrial workplace, sink and hand dryer microbiomes. And me, Aiman bin Aif from AS2567A. There is 6 agenda is very important in this research, which are abstract, introduction, materials and methodology, result and discussion, conclusion and the references. Now we move to the first part, which is the abstract of the research. This study focused on the bacteria and antibiotic resistance on two main surfaces, which are hand dryers and sinks in a pharmaceutical workplace. Samples were collected from two main areas, which are one area the web-led work and another from dry work areas like manufacturing and engineering departments. The study showed that the types of bacteria on hand dryers were different from those on sinks. However, when it comes to the antibiotic-resistant genes, ARGs, the location of the sample had more influence than the sample type. These findings show that human activity, the environment, can affect how resistance spreads. It highlights why environmental surveillance for antimicrobial resistance is important, especially in shared spaces. Now we move to the second part, which is the introduction. First, let's talk about why this study was done. Antibiotic resistance, AMR, is now growing global health problem. It's hard to track how resistance spreads, especially in places without much surveillance. One main cause is the overuse of antibiotic in both humans and animals. Places with high human activity, like workplaces or public toilets, can help spread bacteria and resistant genes. So far, most research has focused on hospital settings like patient sinks and hand dryers. But very few studies have looked at industrial workplaces like factories or labs. Hand dryers are also concerned because they can blow bacteria into the air, spreading contamination further. This study aimed to fill that gap by examining both the microbiome and antibiotic-resistant genes from these surfaces. Now, let's move to the next part, which is the materials and the methodology, which first of all is for the sample collection. For the sampling, researchers took samples from 6 locations, including 3 toilet lanes near wet labs, 3 areas from engineering and manufacturing E&M departments. Samples were collected from the inside of hand dryers and the U-bend of the sink pipes. They used sterile swabs, then placed the swab deep into a buffer solution. For the DNA extraction, they used a microbiome ultranucleic acid isolation kit and an automated machine called Kingfisher Flex. After extraction, DNA was stored at negative 20 degrees Celsius for DNA sequencing. Samples were normalized and prepared using the DNA prep kit. The DNA was sequenced using the Nova Sequence 600 platform. For detection of antibiotic-resistant genes, they used a software tool called RGI, comparing the DNA reads to the CARD database to count and normalize the ARG. For genome assembly, they used SPADAS, Kraken, and Boti software. High-quality genome bins were analyzed further using phylophline to identify which active bacteria carried the ARGs. Finally, for data visualization, they used our software and ClassViz with statistical tests like Kruskal-Wallis and Permanova. We move to the next part, the crucial part, which is the results and discussions. First of all, the DNA extraction and the sequencing. The DNA was successfully extracted and sequenced from 12 samples, which is 6 sinks and 6 hand dryers. The sequencing produced around 130 GB of data with no poor quality reads. And here we can look up for the table on the area distance of the different sample collection. Next, we move to the microbial communities, hand dryers versus sinks. Across all samples, the most common bacterial group was Proteobacteria, followed by Actinobacteria. Sinks had a higher proportion of Proteobacteria, about 84%, compared to hand dryers around 72%. When looking more closely at the family level, each sample had different dominant bacterial families. For example, in one hand dryer, Bucholdicoraceae made up 96% of the bacteria. Overall, there was no single dominant family across all samples, showing that bacterial diversity varied a lot between samples. Next, the second part of the result and discussion. For the antibiotic-resistant genes, ARGs, the common ARGs were against each fluoroquinolines, tetracyclines, disinfectants, and glycopeptides. Both sinks and hand dryers had similar amount of ARGs. However, when grouped by location, E and M areas had more ARGs than lab adjacent toilet areas. This could be due to differences in cleaning practices, user behavior, or type of work done in each area. Then, main takeaways on microbiome vs resistant. Microbiome composition was affected more by the sample, type, sink, and hand dryer. Meanwhile, resistant profiles ARGs were more affected by location, land vs E and M areas. This suggests that environmental factors like cleaning, routine, user habits, and work type can affect to the spread of resistant gene. Thus, as the conclusion, this study shows that the type of surface affects the bacterial population more than the proximity between the surfaces. Even hand dryers and sinks in the same room and very different microbiomes, for RGs, location and human activity play a big role. This research highlights the need for continued AMR surveillance in workplace. It also suggests that design and maintenance of hand washing facilities should consider their role in spreading the bacteria-resistant gene. And this is the references. For this presentation, the main reference I use is the research article by Thompson and colleagues published in BMC Microbiology in 2025. Other supporting references used in the article include studies on antimicrobial resistance, microbiomes in workplace environments, and bacterial spreads from hand dryers and sinks. I think that's all from me. Thank you for hearing me, and feel free to ask any question about this research. Thank you.